So I was explaining you where Sanger's dideoxynucleotide sequencing method would not work. So think about the condition where your template DNA molecule would have a strong secondary structure. Secondary structure, what does that mean? So your template DNA should have been denatured. So it should exist as a single strand DNA in order for a primer to be able to bind and start the replication, right? But however, the template DNA could have a secondary structure such as a hairpin loop. Remember the hairpin loop structure when you talked about the primers? So DNA molecule can be folded up by itself because there is a complementarity existing inside of the single strand. Okay, so think about that scenario when a template DNA, the single strand template DNA has a complementary structure, so it's folding up. Okay, if that happened, okay, let's say polymerase bound to its target site and DNA polymerase grabbed the tail of the primer and is trying to extend, okay? But when DNA polymerase tries to extend that area where the hairpin loop is formed on the template DNA, it's going to stall because the hairpin loop is blocking the advancement of DNA polymerase. So your DNA polymerase will stop its job. So there is going to be a random termination. We call the random termination, premature termination, even without the dideoxynucleotide added to the newly made strand. Okay, so if you have a strong secondary structure on the template DNA, you will see, sequencing is not going to happen. It's going to just go stop, and normal replication will take place. So that's one example where Sanger dideoxynucleotide sequencing would not work if there's a strong presence of secondary structure on your template DNA. Okay. Another possibility is, the secondary structure is made on the replicate strand, for example. Okay. So the replicate strand with the dideoxynucleotide added, when they are separated on a gel, somehow they have those secondary structures. So they are not sorted out based on the sizes. Instead, they are stopped, and they may have different sizes, but since they have secondary structure, hairpin structure, they may show up as the same size, although they have different dideoxynucleotide at the ends. For example, look at that pig right there. You see what, black and green and superimposed together. Looks like there are two deoxynucleotide stopping the replication there. So that is what we're talking about as compression. That's due to the strong presence of secondary structure on your replicate strand. Then it's hard to tell, is this what nucleotide, right? It's hard to identify the sequencing. So those difficulties will be present if we use Sanger dideoxynucleotide sequencing method. Okay, so under those conditions, yes, this chemical-based sequencing method works because this doesn't depend on the DNA replication process. It doesn't matter DNA polymerase, it will be able to move through the DNA template DNA or not, because you're not doing the replication, right? So you can switch the sequencing method to this, strong mutagen-based Max and Gilbert method, and identify the sequence of those areas. Okay, so it's a kind of alternative method. If you have strong secondary structures on your template DNA or on your replicate strand, then you can identify the sequence of those. Okay, so this is the alternative method. Make sense? So again, the comparison of two methods, Sanger's dideoxy method is more rapid and efficient method. Okay, more universal method. However, it is subjected to a stop when there are strong secondary structures on the template DNA as well as on the replicate DNA molecules. Okay, then it's not going to work. And if that's the case, then you may have to use chemical-based sequencing method. Okay, this method is less susceptible to secondary structure problem, and it does not require a subcloning into a vector, or you need to make the denatured template DNA. Okay. So if you are using that chemical-based sequencing method, so how would you be able to visualize the DNA on a gel, electrophoresis? Because you didn't add the fluorescent dye. What is the alternative method of detecting the bands? So back in the old days, we labeled the DNA with radioactive isotopes such as alpha 35 or P32. Okay, these radioactive isotopes were added to your maybe primer or your DNA, to replicate DNA. So they will give you strong signal. So when those replicate products were sorted out, you can visualize the band because you can impose that radioactivity isotope on X-ray film. Then it'll make the signals on the film. Okay, that's how the detection was done. So it's kind of old technology again. Okay, and then another way of doing that, if the radioactive isotope usage is too dangerous because you're exposed to radiation, you can use alternative method. That's chemiluminescence labels, meaning chemical signaling method. There are some chemicals that can generate light energy, and you can use those chemicals to detect the DNA, because those chemicals will be added to your DNA like a primer, and once the primer is attached to a newly made strand, then you can detect those. So that's chemiluminescence based labeling method. Okay, so there are enzymes that can help you to label your DNA with these chemical molecules. Okay, so that's illustrated there. Streptavidin particle is conjugated. So this is DNA. DNA is attached to it something called biotin. Okay, and then biotin and streptavidin can bind strongly. So by doing so, you can label it, and you can attach some enzymes there, and the enzymes can generate the light energy. That's chemiluminescence. Okay. And we already talked about this advancement in the Sanger dideoxynucleotide sequencing method. That is the cyclic sequencing. We already talked about that. We already looked at that, right? So this is a machine that's doing the cyclic sequencing. So you see the little thin line here. That's a capillary gel. So electrophoresis gel will run through there. Okay, so you can sequence up to 1,000 base pair, and that's again kind of an old technology. These days they use something better than this, like a pyro sequencing and other alternative method. So anyway, if we talk about the cyclic sequencing method, yeah, you're going to add a mixture of template DNA and then primers and regular DNA nucleotide, and then dideoxynucleotide, you're going to add those, right, and separate out those newly made strands on a gel. You're gonna detect them. That's how it works, right? DNA sequencing strategies. When you sequence out the nucleotide sequence of the DNA, let's say your gene is located in here in the double-strand DNA of the genome, do you sequence out one of the two strands? Because the other strand is complementary to whatever the sequence you're sequencing out, right? The answer is no. You have to sequence out both to make sure that this sequencing is matching to this complementarity, right? So by sequencing out both strands, you will make sure that what you identified is correct for the accuracy of it. Okay, it's because there are this compression based on secondary structures. There's maybe some ambiguity in your sequencing results. Then that ambiguity could be solved if you sequence both strands. If they are matching sequence you can get, and that's a better result, right? And back in the older days, the PCR technology was not that available. Then you might had to clone your gene into a plasmid DNA, and that recombinant DNA could be moved into a new host cell, and then you grow the host cell in large quantity, and you can make a lot more copies of DNA, and from those copies you can sequence out your DNA. So that was a better approach at that time, right? But these days you have better PCR approach, so the subcloning strategy is not important anymore. It's a kind of old technology, but if people say about subcloning, that's why they had to do that at that time, right? So if you are interested in maybe sequencing out a chromosomal DNA, it can be chopped up into different pieces, and those small pieces can be individually cloned into maybe plasmid DNA, and all those clones could be replicated, and you can sequence one clone at a time, or you can chop up the chromosomal DNA into random small pieces, and all those random small pieces could be sequenced directly one at a time randomly, and then the sequence could be analyzed by a supercomputer. That's how the sequencing, genome sequencing, was done back in 1990s. Again, this could be an old topic, but there's a unique vector. So a vector that is made out of bacteriophage M13. You know what bacteriophages are, right? Those are viruses that can infect E. coli cells. So the unique feature of this M13 bacteriophage is they have a unique genome. Okay, that genome can exist as double-strand DNA or as in single-strand DNA. So that was a unique, I guess, benefit of this M13 bacteriophage genome-based vector. Okay, so if you clone your gene of interest, that will be sequenced out within this vector, bacteriophage M13. The benefit you will get is you can make that into a single-strand DNA without going through the denaturation process, because the bacteriophage, when it infects the E. coli and when it comes out, it can present you a single-stranded DNA genome. Okay, then you don't have to do the denaturation process before you do the sequencing. That is the whole benefit of using M13 vector, and then you clone your gene in there. Okay, so all these explanations are all about that. Single-stranded capacitive M13 will be handy if you clone your gene of interest there and then sequence. Okay, so M13 looks like that. It's got a 6.4 kilobase pair size, and the promoter element is located there, and the terminator is some other area, and you can probably clone your gene somewhere in between there. Okay, and this is probably the life cycle of M13. Let's say this is the E. coli cell, and M13 can insert and inject its genome. So that's an infecting strand. Again, it's a single strand that you see. So it carries a single strand DNA genome, and then inside of the host cell it replicates. It becomes a double-stranded DNA transiently in a very short period of time, and during the replication it makes a lot of copies, and then later on it becomes single-stranded again, and then it packages its old daughter viruses, and then it releases by killing the host cell. So that's the life cycle of a bacteriophage M13. Okay, so it's this unique feature that will be beneficial if you want to clone your gene in there, and then it'll become single-stranded. You don't have to do the denaturation process. Then you can just add primer and then add the DNA polymerase. You can sequence out right away. Okay, so if you are doing a DNA sequencing, okay, so preparing your template DNA is really important, because that's what your sequencing will be performed, right? When you do that, you have to have enough DNA, as well as that DNA should be very clean, because it has a lot of junk like SDS and some other random DNA pieces. Your sequencing is not going to work. You'll never be able to get a clean sequencing result. Okay? So you have to make sure that your DNA will be well cleaned. So if you have some, detergent and things mixed up with your DNA result, sequencing results is not good, then make sure you do a precipitation of your DNA with lithium chloride. Also, you can remove RNA molecules if they are mixed up with your DNA with RNase A treatment. You can get rid of those, and once that's done, you can remove the enzymes by doing a phenylchloroform extraction. Okay, and then after having your DNA, of course your DNA molecule has to be denatured. You can use PCR for that, or you can use alkaline solution treatment. So it's kind of old-fashioned treatment, but it's always good to use the alkaline solution. So, for example, plasmid DNA is denatured using sodium hydroxide, about 2 molar concentration with the presence of EDTA, and then once the denaturation is done, then you can neutralize the DNA by adding, 3 molar sodium acetate at pH 6. That's how typically the denaturation step is done. And then the ratio of primer to your template DNA is also important. Okay, the optimum ratio is about 1:1, so you have to add enough primers. If your DNA strands are all single-stranded, then you can have 1:1 ratio. But if you have double-strand DNA, the ratio should be 2:1. Yeah, that's typical. If you had too much primer, it may create some false priming site, meaning what? Primer is binding at the non-specific binding sites on the DNA template. So that's not good, because you're going to have kind of a shadow or ghost image showing up on your DNA sequencing results. Okay, so do not add too much primer. And then, yeah, primer binding condition, like annealing temperature condition, is also important. So you need to figure out what that annealing temperature is. You're familiar with this equation, right?